Hello everyone, how are you doing? I hope everyone is doing good. So welcome to another video of molecular basis of inheritance. In this video, we will be talking about DNA fingerprinting. That is the application part of the use of DNA that can be extended to various techniques. One of the techniques is DNA fingerprinting. Now, another name for DNA fingerprinting is also DNA profiling. You might have heard this terminology in school also, or otherwise also. That is what happens in DNA fingerprinting. That is identification of an individual based on their DNA sequence, not on the fingerprint. So basically, this is a misnomer over here. Actually, it's DNA profiling. So identification of an individual based on his or her DNA sequence. So we all know that everyone's DNA sequence is unique. Okay, the pattern is unique. Though you might be amazed by knowing that we are similar to one another by 99.9%. Only that uniqueness or difference comes comes only by this small percentage of number. That is, we are different from one another, or we are unique from one another only by 0.1%. Okay, so let's just begin with DNA fingerprinting, what it is. So basically, it's a method wherein you can identify an individual. So basically, DNA fingerprinting is used in forensic sciences, in crime places, in parentage dispute. Okay, these places where DNA profiling is used. Okay, so to identify an individual from a sample of DNA by looking at his or her unique patterns of the DNA, by looking at unique patterns in their DNA. So we are looking only at that 0.1% uniqueness, which will be different from mine than yours, from yours to your, everyone on this planet Earth. So, so identification is done on that basis. Okay. So we are going to see the technique. This was developed by Alec Jeffrey in 1985. Okay, he was working with some organism, bacterial organism, and then he came up with this technique. So DNA of an organism carries some non-coding sequences called VNTRs, or called repetitive sequence. Okay, we just mentioned about the uniqueness, right? This uniqueness is because of this. This uniqueness is because of this repetitive sequences which you will see in every individual, and this repetitive sequences will be different in different individual. That uniqueness is because of this. So my repetitive sequence will be completely in a different will be completely unique than yours. Okay, they're also known as mini satellites, or they're also known as, the technical term is actually variable number of tandem repeats. So in an individual, you will see this particular sequence occurring again and again throughout their genome, throughout their chromosome sequence, throughout their genome. Okay, so variable means that number will differ. In, let's say, my it might be 1,000, or in it might be some 1593 or something like that, for example. Just giving you an arbitrary number. Definitely they are more than that, more than that. So variable number of tandem repeats. Tandem means again and again you will find them in your genome, and that sequence will be completely different from the completely different, completely unique from any other person on the earth. Okay, so on this basis, so when we are talking about identifying an individual based on the sample of DNA, so we are identifying the VNTR. We are recognizing the VNTR of different person. Okay, on that basis, we identify the individual, which are non-coding sequences. So they don't code for any any protein or any enzyme or any other metabolic molecule. They are just present for identification purpose. Okay. So this is a general understanding about what is DNA profiling. and where it is, on what basis do we identify DNA. That is that 99.9% we are similar, only 0.1% we are different, and that difference is because of this VNTRs. Okay, so number of these repeats is very, very specific and unique from person to person. So we are going to understand briefly about the topic of DNA fingerprinting. Okay, so these are the steps that we are going to discuss. We are going to learn briefly. That is isolation of first what will happen in the technique. This is a brief understanding. We are not going to the picture at the moment right now because we're going to discuss each and every step in detail, and then we will take this picture again up at the end. So isolation of the DNA, that is wherever you find the DNA, you take the DNA and isolate it. You separate the DNA from the rest of the material of the cell. Then you amplify it, basically increase the number of that sample because the DNA obviously will be in very less concentration or less amount. Then digestion of the DNA, breaking of the DNA into smaller parts. Then there are three techniques which you see over here: gel electrophoresis, blotting, and autoradiography. Finally, is the taking picture. Okay, the image is created of that particular profile. So we are going to take this up one by one. We will begin with isolation of DNA first. Okay, so let's begin. So, so as you can see, the first step over here, isolation of the DNA. How is it done? To memorize it or learn it in very detail, uh, just a brief understanding also will be enough for you to write in the exam under the heading of isolation of DNA. So as you can see in this string, you can see this is the DNA. Okay, precipitated DNA. So what happens is first you get the sample. Okay, and you take the suspected samples of DNA, either in the form of, usually it is in the form of blood only. From the blood you extract the DNA. So how this DNA is extracted from the blood? You take the cell, you break as in you lyse it using some detergents, some solution, which breaks the plasma membrane. And when the plasma membrane are broken up, all the contents which are there in the cell will come out, right? So if the cell is there, you have the nucleus, you have the cytoplasm and all this thing. When you break this plasma membrane, okay, everything will come out in the solution. So now in the solution there will be proteins, there will be RNA, there will be DNA, there will be some other molecules, etc., which you are not interested in. So you will use enzymes, other enzymes to remove mostly protein and RNA. Okay, so you are not interested in these two important parts of the cell. You are left with DNA, and then what you will do? So you use all this, and then there is a solution. You have to separate now the all broken parts, that is the cell debris. You have to separate, and the DNA which is left intact in the solution. So there is a centrifuge machine, okay, which rotates at very high speed. So you put the test tube in that machine, centrifuge machine, and the DNA will come in the liquid part, and the heavier material will settle it down. Like just you can see in the picture, the liquid part will float up, and the heavy material, the red blood cells, all the broken parts, cell debris, will be settled at the bottom. And you transfer this supernatant to another part, another test tube, and you add alcohol in it. Once you add alcohol in it, you get the DNA. Okay, so the DNA can be removed out from the test tube with the help of a glass rod or with the help of any tool. Okay, so the first step in the profiling is first you isolate the DNA. So this is how you isolate the DNA from other components of the cell. Then what you do, you have the DNA, you will take the DNA and you will uh, break it into fragments. Okay. Now sometimes the DNA sample will be very very less, so you do a PCR technique which is known as polymerase chain reaction technique that we will see in another chapter when we discuss biotechnology, that is amplification of the DNA. So if the DNA, the amount of DNA that you get from the isolation technique is very very less, the quantity of DNA is very less to carry out the further test, further steps, so you will amplify the DNA. Basically, you will increase the amount of DNA through a technique known as PCR, polymerase chain reaction. This we will see in the next chapter. Okay. So you will basically, you are doing just amplifying, increasing the amount of DNA so that you have sufficient amount of sample for the techniques, the further steps to be carried out in DNA fingerprinting. Now, after amplification is done, now you will digest the DNA. So you have the whole long linear DNA. Okay, you will break it up into smaller fragments. You will break it up into smaller fragments. Remember, DNA profiling is done on which principle? On the principle of VNTRs. Okay, that is, you are trying to identify the variable number of random repeats in that particular sample. Okay, and this VNTRs, this repetitive sequence will be unique, will be specific from person to person. So this breaking is done by enzymes. Which enzymes? You need to know the name: restriction endonucleases. Nucleases, restriction endonucleases. Okay, they will break up the DNA into smaller fragments. So they cut the DNA into smaller fragments. Okay, having variable lengths. Okay, so this phenomena is known as RFLP. So once you take your DNA and you add the enzyme, you get small fragments which will have your VNTRs. This particular phenomena, okay, is known as restriction fragment length polymorphism. Right? So once you get these smaller fragments, and in that you will have basically you have uh, chopped the whole DNA into variable lengths. Okay, some smaller, some bigger, some medium size, etc. Okay. So DNA profiling. So the next step will be, now you have a mixture of these fragments in your test tube. Now you will separate these fragments based on their size, smaller size, medium size, bigger size. Okay, this separation is done by a technique known as gel electrophoresis. As you can see the setup over here. So this is a technique. You have this setup, rectangular box-like structure, which is in this you have the gel, you have all the required solution, buffer, and all that stuff. You see the small test tube over here. This is your sample containing the chopped DNA. Okay, the whole DNA was chopped up into smaller fragments. Now you will separate it and you will try to see if you have got the VNTRs or not in the later stages. Okay, and now this setup is run under electricity. The name itself is suggesting electrophoresis. What is electro? Electro means under the influence of electricity, movement of the DNA. The movement of the DNA sample under the influence of electricity. So this is what we call it as electrophoresis. Okay, this is a technique in itself. You just need to know the basic of it. There's a lot more to know about electrophoresis, but right now at your level only, just to know at this step. Okay, a brief understanding about what electrophoresis is doing in this DNA fingerprinting. So now here you see, you take the sample, you put the sample, you load the sample in these small places which is created, which are known as well, and you apply electricity, you apply a power over here, and you allow these fragments, these DNA fragments of varying length to separate. So obviously the longer molecules will remain behind and the shorter molecules, DNA molecules, will move forward. Yes or no? So longer molecules means high molecular weight. High molecular weight, the bigger fragments. of the DNA will be left behind, and the smaller fragments, shorter fragments, which are having low molecular weight, will run ahead. So they will be in the line, like it's a race, and they will reach over here. So DNA being negatively charged, it will run towards the positive terminal. You can see positive terminal over here and the negative terminal over here. Okay, this you are aware of cathode anode biochemistry, and you should be knowing this working. Okay, so DNA being negatively charged will run towards the positive terminal, that is the anode positive terminal. Okay, so this is how the fragments will be separated by gel electrophoresis. Now, once you get a separated band, you will transfer it on a paper. You will treat it with some solution. Here you will treat it with sodium hydroxide. Okay, because the bands are what, double stranded. So you need to identify these VNTRs. So one of the fragments you will remove. You will just you will be left with only single stranded bands. So you'll remove the other bands because you will now add your own band, probes, your own single stranded probing for identification. So you will treat this gel with solution, alkali solution, that is sodium hydroxide, to separate the bands in the gel into single stranded. Okay, DNA is double stranded, you will make it single strand first. Then once it is single stranded, you will transfer it to the paper or a membrane. Okay, transferring the separated single stranded DNA fragments to a synthetic membrane. Either it can be a nitrocellulose membrane or a nylon membrane. It's like a small paper, like, not your filter paper, but very, very thin membrane. And you will fix the DNA fragment onto that membrane. So from the gel, it is transferred to a paper or to a membrane. Okay. Now, once it is transferred to the filter paper, this is dipped into a solution of radioactively labeled single-stranded DNA probe, because you need to identify where are the repetitive sequence, what are the VNTRs, and how one sample is unique from the another sample. So for that identification process, for that imaging process, you will use your synthetically made radioactive labeled, because this radioactivity will help in taking photo, in taking picture of this band into a software on a computer screen. Okay, so for that purpose, you converted double stranded into single stranded, and now to that left single stranded, you will add your radioactively labeled single stranded DNA probe. So binding will take place now. Okay, and the DNA probe will bind with the complementary sequences. So wherever they find complementary sequences, the probe which you added, which were radioactively labeled, they will go and bind depending upon the complementary sequences onto that membrane. Okay, and now they are in the form of hybridized DNA. So like these are the fragments, double stranded. Yes. So this is the original fragment from the sample, these strands, let's say, and these strands are your radioactively labeled strands. Okay. So now they are what, hybridized. Na? One strand is from the sample, and the another strand which got bind due to complementary pairing, they created a hybridized DNA. So this is now easier to detect, to obtain an image or to obtain a photograph, so that it can be processed and viewed in a software on the computer screen. Okay. So now the filter paper is washed to remove the unbound probe. So whatever remaining probe is there, that will be washed and that will be removed. Right. So this is how you will see. So if, let's say, this is the sample of a person, DNA sample of a person you took from the crime spot, wherever the murder happened or whatever happened, crime spot, and you did DNA fingerprinting, all the steps that we just discussed: DNA isolation, amplification, restriction, endonuclease digestion, then separation, then blotting, transferring, and then the photography now that you will take. And took samples from 3 persons that you were suspecting to be the person who committed the crime. Okay, so you then match the crime scene, you match this profile with the suspected profile. So the hybridized DNA that you got is now photographed onto an X-ray film. Okay, like you take an X-ray film and you obtain an image by this technique known as autoradiography. You just have to know this point, that's it. Autoradiography, if you are interested, you can read more about it on the Google and understand this technique. Basically, this technique is used to obtain a photograph of the uh, bands that you got on the nitrocellulose or the nylon membrane. Okay, so you obtain a photograph onto an X-ray film by this technique, autoradiography. Okay, because you use radioactively labeled probes, and then the image in the form of dark and light bands you can see of the hybridized DNA is which you obtain is called the DNA fingerprint. So this is your DNA fingerprint that you obtained for the let's say you took 4 samples, one which was at the crime spot, then 3 samples from 3 persons that you suspected that could be the criminal, and if you see this is matching with suspect 2. This is matching with suspect 2. You can see the bands exactly matching. The length, the place, and the position and the thickness, everything is matching. So in this case, you can say the suspect who did the crime. So this is all about DNA fingerprinting. Okay. So this is the summarized part. You take the sample, DNA is extracted, isolation of the DNA, then you cut the DNA, smaller fragments. The fragments are separated into bands by electrophoresis on a gel. Then from the gel you transfer it onto the paper or a membrane, nylon Cabanava membrane or nitrocellulose membrane. This technique is known as southern blotting. Okay, just for your information, southern blotting. Then you convert a double-stranded DNA into single-stranded DNA, and then you use your radioactively labeled probe. Then it will bind to the complementary sequences, and you wash the excess off, the unused part, wash it off, and now you get this on a paper. Okay, at this stage the radioactivity probe is bound to the DNA on the membrane, and now this paper is exposed to X-ray film. Then to autoradiography. Okay, X-ray film is placed next to the membrane to detect the radioactive pattern. Right, and then the X-ray film is developed like in earlier days how photo images were developed in same manner. Okay, so the X-ray film that you get now from the paper you transfer it onto an X-ray film. Okay, then this film is developed and made visible the patterns, which is now your fingerprint, DNA fingerprint. You see the bands over here. Okay, so this is how the DNA fingerprinting steps are done. So where can you use DNA fingerprinting? Where you want to identify the real genetic mother, okay, or the father and the offspring. You want to identify the real mother of the child, or the real father of the child, like that. Okay. Then, if there is any problem or arguments or disputes in paternity, maternity, DNA fingerprinting can be identified to do that. So individuality, paternity, maternity dispute, human lineage. When we are doing phylogenetic analysis studies in genetic experiments and studies, there also DNA fingerprintings are used to various hereditary diseases. Can be studied, analyzed using fingerprinting technique. So it is used to identify genes connected with hereditary disease. Okay, and one example we just saw about crime. that is in forensic sciences. So that's the most common application of DNA fingerprinting. So that's all regarding DNA fingerprinting, one of the very important topic which is asked in exam. So go through the video. If you have any doubt, you can ask me. So that's all for this chapter. We are done with it. Keep revising. Thank you. Take care. Bye bye. We'll begin with the next chapter in the next series of recordings.